Hello classmates, in this lesson we will talk about glomerular filtration. Each single nephron contains the structure called the renal corpuscle. The renal corpuscle actually consists of two different structures, the Bowman's corpuscle and the glomerulus. The glomerulus is a network of capillaries, and the endothelium of these capillaries is porous, so that they allow loss of fluid, waste products, ions, glucose, and amino acids to pass from the blood into the corpuscle. But they block out bigger molecules like blood cells and proteins. So they stay in the blood and exist through the afferent arterioles and peritubular capillaries. About 20% of blood gets into the space of Bowman's corpuscle through the filtration layer. Now all the stuff that gets squeezed out of the blood into the glomerulus is called filtrate. We use the micropuncture technique and found the filtrate is protein-free. The glomerular capillaries is relatively impermeable to proteins. The concentrations of most constituents of the glomerular filtrate are similar to those in the plasma. The G-alpha means how much blood passes through the glomeruli every minute. It was affected by coefficient of filtration and effective filtration pressure. Filtration fraction means G-alpha as percentage of total renal plasma flow that passes through both kidneys. It's about 19%. The glomerulus contains two types of specialized cells. Lining the capillaries in the glomerulus is endothelium cells. The Bowman's capsule contains podocytes. They allow loss of fluid, waste products, ions, and glucose to pass from the blood into corpuscle. But they block out bigger molecules like blood cells and proteins. As we can see, the filtration membrane is made of capillary endothelium, basement membrane, filtration slits of podocytes. The endothelium cells are basically involved with filtration. They contain tiny pores called fenestrations. The podocytes contain very small slits. They are with molecular size selectivity and electrical charge selectivity. It is impermeable to substances with molecular weight more than 69,000 or effective radius greater than 4.2 nanometer. Besides, it repels negative charged substances. This table shows the filtratability of substances by glomerular capillaries based on the molecular weight. Note that electrolytes such as sodium and small organic compounds such as glucose are freely filtrated. As the molecular weight of the molecule approaches that of albumin, the filtrability rapidly decreases, approaching to zero. The net filtration pressure represents the sum of hydrostatic and colloid osmotic forces that either favor or oppose the filtration across the glomerular capillaries. These factors include hydrostatic pressure inside the glomerular capillaries, which promotes the filtration. The hydrostatic pressure in Bowman's capsule outside the capillaries, which oppose the filtration. The colloid osmotic pressure of the glomerular capillaries plasma proteins, which oppose the filtration. And the colloid osmotic pressure of the proteins in Bowman's capsule, which promotes filtration. Under normal conditions, it is considered to be zero. According to data from dogs and rats, scientists estimate net filtration pressure is about 10 mm mercury. Normally, about 20% of the fluid in the glomerular capillary filtrates into Bowman's space, thereby concentrating the plasma proteins that are not filtered. Increase in the filtration factor promote the plasma colloid osmotic pressure rising along the glomerular capillary. Decrease in the filtration factor have the opposite effect. Capillary blood pressure is one of determinants of GFR. It varies with arterial blood pressure and resistance of efferent and afferent arterioles. When blood pressure is among 18 to 180 mm mercury, GFR is relative constant. When blood pressure is among 18 to 180 mm mercury, GFR is relative constant. When blood pressure is less than 50 mm mercury, GFR is almost zero. Blood colloid osmotic pressure is another determinant of GFR. Under normal conditions, it doesn't change significantly. When intravenous infusion of a large volume of saline or albumin loss increases, plasma protein concentration decreases, which increases the GFR. Moreover, capsular pressure, renal blood flow, and filtration coefficient could also affect the GFR. Okay, this lesson is over. Thanks for your listening.